Welcome to this explainer. Today, we're skipping the small talk and diving straight into literally one of the most mind-boggling physics puzzles in all of biology. We're looking at the ultimate spatial crisis, comparing how two fundamentally different domains of life, simple bacteria and complex eukaryotes, tackled the exact same, seemingly impossible problem.
Just think about this staggering mathematical impossibility for a second. An E. coli bacterium contains a linear length of DNA that is 500 times longer than the entire cell itself. Imagine trying to fit a piece of string that's half a kilometer long into a box that's just 1 meter wide. It's basically like trying to stuff a massive parachute into a tiny backpack. So that brings us to the ultimate question we're answering today. How does life manage to compress massive amounts of genetic code into such unbelievably tiny, microscopic spaces? And more importantly, how does it pack this DNA so tightly without tangling it into an absolute unusable knot? We all know what happens to a pair of headphones left in a pocket for 5 minutes. So how exactly does biology pull this off? Here's our roadmap for today. We'll hit the ultimate packing problem, look inside the simple nucleoid, check out the eukaryotic membrane vault, dive into histones and nucleosome beads, trace the chromatin telephone cord, and finally compare the two domains. Let's get into it.
Section 1. The ultimate packing problem. To really appreciate what cellular architecture does, we've got to understand the sheer scale of the space constraint that simple physics imposes on life. There's a stark contrast here. Both simple bacteria and complex human cells are dealing with a massive spatial crisis. I mean, a tiny 3 micrometer E. coli has to wrangle 1.5 millimeters of DNA. And a human cell? It's packing a whopping 2 meters of linear DNA into a nucleus that's a mere 10 micrometers across. It's wild. And as we're going to see, they solve this exact same spatial problem in completely different ways. Moving to Section 2, inside the simple nucleoid. Let's look at how prokaryotes, the single-celled organisms like bacteria that keep things relatively simple, handle this. Since they don't have a membrane-bound vault, bacteria organize their DNA right in an open region of the cytoplasm. It functions a lot like a highly efficient, minimalist filing cabinet. They're working with a single circular DNA molecule and a more modest library of about 500 to 4,000 genes. But hey, even with a smaller gene count, 1.5 millimeters of DNA still isn't going to fold itself. So what's their secret? Bacteria actively twist and fold that circular DNA into roughly 400 independent, negatively supercoiled loops, and they get some specialized help to pull this off. They use a protein called integration host factor, or IHF, to sharply bend the DNA. At the same time, SMC proteins act like tiny biological zip ties. They literally link bits of DNA together to pin the whole shebang into a compact, organized nucleoid. All right, Section 3, Eukaryotes and the membrane vault. Having seen the minimalist approach, let's see how complex life forms handle a drastically larger genetic burden. Now, eukaryotes have a much heavier lift, managing anywhere from 6,000 to 30,000 genes spread across multiple linear chromosomes. Unlike the open floor plan we just saw in bacteria, these complex cells evolved a dedicated, highly secure compartment. Yep, the nucleus, wrapped in a protective lipid membrane. But of course, just building a vault doesn't magically solve the physical problem of coiling 2 meters of DNA into it. Which brings us to a brilliantly simple concept. If you've ever tried to store a garden hose, you know it's a million times easier if you actually coil the thing first. The cell can't just shove the DNA inside, it has to be deliberately coiled. And that takes real work. The cell uses specialized proteins to actively provide the energy, mostly through electrostatic interactions, to really fold that DNA into submission. That leads us to Section 4, histones and nucleosome beads. To perform this incredible feat of molecular coiling, eukaryotic cells rely on an ingenious, microscopic spooling system. Check out the brilliant use of physics in this basic packing unit, the nucleosome. Because DNA's phosphodiester backbone is negatively charged, the cell wraps it tightly around a core of positively charged proteins. It's utilizing pure electrostatic attraction to keep the DNA perfectly organized and powerfully gripped. These positive proteins are called histones, and they are highly conserved across species. The cell takes 2 each of 4 core histones, H2A, H2B, H3, and H4, and builds an 8-part spool called an octamer. Then, about 200 base pairs of DNA wrap around this spool in a left-handed manner. That left-handed wrap is actually absolutely crucial because it leaves the DNA negatively supercoiled, making it much easier to unwind later.
Finally, you've got a fifth protein, histone H1, acting almost like a microscopic piece of tape that clips the outside of the structure to lock the DNA onto the spool. On to section 5, the chromatin telephone cord. Now that we have our nucleosome beads, the cell stacks these spools into increasingly complex structures to save even more space, creating a dense material known as chromatin. So how does this all stack up? First, we get the 10-nanometer fiber, which literally looks like beads on a string. Then, those beads twist themselves into a 30-nanometer helical fiber. If you remember those old-fashioned spiral telephone cords, it's exactly like that. But it doesn't stop there. That telephone cord gathers and loops back onto itself along a protein scaffold, creating massive 300-nanometer looped domains. Eventually, this forms the highly condensed chromosomes you see right before a cell divides. When we look at actual electron micrographs, we can witness this exact progression happening. First, there's that incredibly tight 30-nanometer helical fiber, and then you see how that fiber erupts into massive looping domains. This right here is the predominant form the DNA takes inside the nucleus during interphase, the cell's normal resting state. Now, what's really fascinating is that this extreme packing isn't just for long-term storage. It's highly dynamic. The cell is constantly shifting its DNA between two main states. Euchromatin is loosely packed and kept near the center of the nucleus. This is where your active genes live, ready to be transcribed. Heterochromatin, on the other hand, is densely packed, shoved off to the outer edges of the nucleus, and genetically silenced. Why does this matter to us? Because simply by altering how tightly the DNA is spooled, the cell literally controls exactly which genes are turned on and which are turned off. It's a true game changer. Finally, section 6, comparing the two domains. We've explored the primary strategies for combating the cellular packing problem, so let's put them head to head. Comparing these features really sums up our whole journey today. Both systems solve the exact same physics problem, just with totally different toolkits. The prokaryotic nucleoid relies on negative supercoiling and bending proteins to manage a modest circular genetic code right out in the open cytoplasm. On the flip side, to handle a massive linear genetic code of up to 30,000 genes, the eukaryotic nucleus evolved a highly secure, membrane-bound vault and an unbelievably complex histone spooling system. And, this leaves us with a pretty fascinating thought to close on. As life continues to evolve and genomes get ever more complex, are we eventually going to hit a fundamental physical limit? Like, how much more tightly can we realistically pack the blueprints of life before the molecular machinery just can't read the code anymore? It's wild to think about. Thanks so much for joining me on this explainer. Keep wondering, and we'll see you next time.